Okay, I think we can go ahead and get started. Good morning, evening, afternoon to everyone. Thank you for joining today's webinar to learn more about the environmental enteric dysfunction, WASH, and nutritional status of women, infants, and young children. Findings from Uganda, Sierra Leone, and Nepal. My name is Yaritza Rodriguez, and I'm a project coordinator at USAID Advancing Nutrition, and I will be your MC today. As more attendees are joining the webinar, I'll begin by going over some of the housekeeping items. Then I'll hand it over to Dr. Patrick Webb, Director of the Innovation Lab for Nutrition, who will give a brief background on the lab and its research. And then he will begin the webinar by introducing today's moderator, Dr. Christopher Duggan. I would like to direct all attendees to a few functions on this Zoom call, Zoom webinar. At the bottom of your screen, you should see a chat icon and a Q&A icon. Next slide, please. So please, like I said before, use the chat function to engage in relevant conversation with other attendees and to introduce yourselves. Panelists will respond to questions in the Q&A box, at which point the panelists, as they are able. We have allotted the final 20 minutes of this webinar for question and answer, at which point the panelists will respond to any remaining questions from the audience. If you're experiencing technical difficulties, send a message in the chat box directly to panelists or a private message to AV support so that our technical support staff can work with you to help resolve any technical issues. This webinar is being recorded and will be made available on the Innovation Lab for Nutrition and USAID Advancing Nutrition websites. You can also register for upcoming webinars and view previous recordings and slide decks on our websites. I will repeat these technical housekeeping items in the chat throughout the webinar as people may be joining in at later times. The moderator for today will be Dr. Christopher Duggan. Dr. Duggan is a pediatric gastroenterologist and nutrition physician at Boston Children's Hospital, a professor of pediatrics at the Harvard Medical School, and a professor at the Harvard T.H. Chan School of Public Health. For the past 25 years, Christopher Duggan has been performing clinical trials in the fields of pediatric nutrition, gastroenterology, and global health. Dr. Duggan and colleagues have evaluated the efficacy of micronutrient supplementation in infants and young children born to women with or at risk of HIV infection. But before handing it off to our moderator, I would like to introduce the director of the Innovation Lab for Nutrition, Professor Patrick Webb, who will give a brief introduction on the Innovation Lab. Dr. Webb, over to you.
Thank you so much, and welcome to everyone to this, the fourth, I believe, webinar in a series this fall, featuring highlighting findings from the Feed the Future Innovation Lab for Nutrition's research around the world. Next slide, please. We have been conducting for the last 10 years in collaboration with many partners, a series of studies of many different kinds in many different countries, focused primarily in sub-Saharan Africa and South Asia, all wrapped around the themes of trying to understand the ecological, economic, and human biological mechanisms that help translate agriculture through diets to improved health and nutrition. So we've been looking at multi-sector programs and the governance of policies and implementation issues, but also a range of biological mechanisms, human mechanisms, sometimes related to mycotoxins and food safety, so foodborne diseases, but today dealing with what goes on in the gut. Next, please. We do this work with a whole range of partners. Chris Duggan, who will chair this particular webinar, comes to us from one of our core partners from Harvard. We have Johns Hopkins, Tuskegee, and Purdue also as core partners, but as you can see, many US, global, and national host country partners doing many different things, but without whom we could not have engaged in the rigor and the scale of activities that we've been able to for this particular series of studies. Next, please. We have a lot of specific collaborators for the studies that are going to be presented today. You're going to hear from findings relating to work in Nepal, Uganda, Sierra Leone, but also there are global partners as well. These range from field workers collecting urine and feces and trying to retain a cold chain all the way to labs, either in country and outside the country, trying to get an understanding of the importance of what is going on inside the gut in terms of permeability, the absorptive capacity for nutrients, to be able to translate what happens in a diet to what happens physiologically in terms of health and nutrition. Next slide, please. I'm going to be, the research theme that we're talking about here relates, as I said, to biological mechanisms. Next, please. And we have a stellar lineup of really important findings relating to the three countries I mentioned. And Chris, as moderator, could not be a better person. He's been involved with us from the beginning. He's a pediatric gastroenterologist. He leads the Center for Nutrition at Boston Children's Hospital, but he's also the medical director for the Center for Advanced Intestinal Rehabilitation. So in terms of expertise, as well as being all-round great guy, we are very honored to have Chris take on leadership of this webinar. So over to you, Chris.
Thank you, Patrick, and thank you, Yuritsa, for the double introduction. It was great, and it is my pleasure to moderate a really exciting session today. And I'm excited to see nearly 300 participants already signed on, and hopefully more as the seminar gets going. I would like now to introduce the panelists. Jackie Lauer is a former Peace Corps volunteer and former postdoc with my program, who is now a public health nutritionist and clinical assistant professor at Boston University. Dr. Lauer's research focuses on environmental contributors to poor growth and neurodevelopment among infants and young children in low resource settings, including environmental enteric dysfunction, which we'll talk about extensively today, as well as aflatoxin exposure, which was the topic of our previous seminar. We're also joined by Akriti Singh. In August, Dr. Singh successfully completed her thesis defense to receive her PhD from Tufts University's Friedman School of Nutrition Science and Policy. Dr. Singh's research focuses on determinants of maternal and child undernutrition in low and middle income countries, including diets, body composition, EED, gut microbiota, and water sanitation and hygiene. Again, the topics of her lectures today. And finally, Dr. Shibani Ghosh, who is the research associate professor at Friedman School of Nutrition Science and Policy, who is working closely with Patrick in her role as associate director of the Innovation Lab for Nutrition. She has extensive experience working in the Middle East, Sub-Saharan Africa, and South Asia. Dr. Ghosh's research interests are in understanding the role of agriculture in improving nutrition while ensuring health, assessing the diet and non-diet determinations of nutritional status of infants and young children, and testing interventions aimed at improving maternal and infant nutrition and growth. So a stellar panel, and we'll start with you, Jackie. Thank you.
Great, thank you, Chris, for that introduction, and warmest welcomes to everyone in attendance for this talk, which is primarily on environmental enteric dysfunction. Today, I'm going to be presenting findings from a few studies that we conducted in Uganda during my time both as a PhD student at Tufts University and during my postdoc at Boston Children's Hospital, where I worked closely with Dr. Duggan. And because I am the first speaker in this webinar, I plan to begin my talk by giving a brief background into environmental enteric dysfunction. Next slide, please. And I have no disclosures in relation to this presentation. Next slide, please. So as highlighted by the 2013 Lancet series, there were and arguably continue to be major problems in our approach to how we address stunting. The report found that if our 10 best nutrition-specific interventions, that is dietary interventions, which are listed here on the slide, were to reach 90% of their target population, the prevalence of stunting would be reduced by only 20%. From these findings, many of us concluded that non-dietary contributors to stunting must have a much bigger role than we previously thought. And we ought to be looking a little more closely at the environment, at disease, and specifically at environmental enteric dysfunction as a driver for stunting. Next slide, please. So looking at the UNICEF malnutrition framework, which depicts pathways to poor nutrition and mortality for young children, the role of inadequate dietary intake has arguably been a disproportionate focus for the nutrition community. And conversely, the disease pathway has been defined too narrowly. While previously thought to include mostly diarrheal disease and other common childhood illnesses, I would propose to reframe this as more of an inflammation, immune suppression, and growth hormone suppression pathway. And there is budding evidence on the drivers of this pathway, which may include not only environmental enteric dysfunction, but also exposure to toxins, including aflatoxin exposure, which was touched on in last week's webinar, and exposure to other environmental contaminants, including air pollution and pesticides. Next slide, please. So the following are the questions I tend to focus on in my research, and the ones I will touch on for the remainder of my presentation today. So to begin with, how should EED be assessed? In other words, what biomarkers should we be using? Also, what are the underlying contributors to EED? What is the relationship between infant and child EED and poor growth outcomes and micronutrient deficiencies? And what is the relationship between maternal EED during pregnancy and poor birth outcomes? Next slide, please. So again, what is EED and how is it assessed is how I'll begin my talk. Problematically, EED still has no universal case definition or diagnostic criteria. Instead, we characterize the condition by changes to both the structure and the function of the small intestine. And these changes include things like blunting of the villi, reduced epithelial surface area and absorptive capacity, altered mucosal barrier integrity, and intestinal and systemic inflammation. So you can see two intestinal biopsies depicted here on this slide. One is showing a healthy intestine on the top and one with EED on the bottom, where you can clearly see the blunted and inflamed villi and naturally reduced absorptive capacity. And it is believed that these changes to the small intestine develop throughout infancy as the result of exposure to chronic exposure to enteropathogens due to living in conditions of poor water and sanitation and hygiene, or what we refer to as WASH. So in fact, it is believed that EED is almost practically ubiquitous among infants and children living in such conditions. Next slide, please. So this schematic depicts the two primary pathways by which EED can lead to stunting in children, or perhaps to low birth weight in the case of maternal EED. Infants and young children living in conditions of poor WASH frequently ingest enteropathogens, which can affect the structure and the function of their small intestine, leading to intestinal inflammation and villus atrophy. This, in and of itself, can reduce the absorptive capacity of the small intestine and ultimately result in poor linear growth. Additionally, it can lead to what we call microbial translocation, or the passage of bacteria across the intestine, which ultimately leads to systemic inflammation. Therefore, it is believed that these children with EED are in a state of constant low-grade immune activation. So not only does the body have to divert nutrients towards fighting this inflammation, but it is also thought that systemic inflammation can suppress the growth hormone, insulin-like growth factor 1, and insulin growth factor binding protein axis, which is essential for normal linear growth. Next slide, please. So EED was actually first recognized in the 1960s and 1970s in a series of studies, of which I'm specifically mentioning two. The Lindenbaum study was the first to identify the condition in returned Peace Corps volunteers. And later it was discovered that the structure and function of the intestine returned to normal after moving to a sanitary environment. The conclusion being that EED is reversible. The Chaco study was a necropsy study, which added to the literature by confirming that infants are not born with EED, and therefore it is an acquired condition, likely from the environment. Next slide, please. Arguably, however, EED did not become noteworthy until the 1991 Lund study from the Gambia. The objective of this study was to explore the relationship between intestinal disease and poor growth in Gambian children who were 2 to 15 years old. The study found that up to 43% of observed growth faltering could be explained by EED. Notably, however, this 43% has not been replicated, but a number of studies since, including our own, have also linked EED to poor growth outcomes in children. Next slide, please. The Lund study assessed EED using the lactulose-mannitol, or the LM test. In an ideal world, intestinal biopsies would provide the best method for assessing EED. However, these are impractical and often unethical in field-based settings. In lieu of biopsies, historically, the most commonly used proxy biomarker has been the LM test, though that has been changing more recently. For the LM test, participants are asked to drink a solution containing 2 different sugars. One is a small monosaccharide, or the mannitol, and the other is a larger disaccharide, or lactulose. And urine is collected over the course of approximately 4 to 5 hours. In a healthy gut, the mannitol would be readily absorbed, and the lactulose would not, as it is too big. In EED, however, mannitol is not so readily absorbed because of the blunted villi, and the lactulose would leak out because of the increased permeability of the gut. Therefore, a high LM ratio is thought to be indicative of the condition of EED. Next slide, please. So the LM test, despite being considered somewhat of a silver standard for assessing EED, is fraught with limitations. First, it is time consuming, in that the test can take upwards of 5 hours or even longer if we have a stubborn child. It is burdensome on both participants and on research staff. It's expensive to administer and to analyze. It often has a high rate of test failure due to things like spilled urine or contamination of stool. It lacks formal evaluation studies. It fails to capture a number of the key domains of EED, especially the inflammation component of the condition that we think is so critical. And it has proven to be inconsistently correlated with EED symptoms and growth outcomes in young children. Next slide, please. So as a result of the limitations of the LM test, numerous biomarkers have been proposed across a range of biological specimens, especially various serum and stool markers, many of which are listed here on this slide. These proposed markers assess the various domains of EED, previously mentioned, including things like intestinal permeability, microbial translocation, and inflammation. Notably, the ones that Chris and I have used a lot are microbial translocation markers, specifically the anti-flagellin and anti-lipopolysaccharide, or LPS, immunoglobulins. Next slide, please. One thing to note, however, is that correlation studies looking at panels of these biomarkers have shown little correlation among them, particularly across different domains of EED. And overall, this is considered to be a big limitation of most EED research at the moment, as it is believed that infants and young children can experience EED differently, especially with regard to the severity that each child experiences in terms of the different domains of the condition. Next slide, please. So now that we've looked at how EED is assessed, I want to turn to what are the underlying drivers of EED. As we mentioned, it is believed that EED results from the chronic ingestion of enteropathogens and toxins due to living in poor wash conditions. Next slide, please. However, with regard to the actual evidence, this is actually still considered somewhat of a hypothesis, primarily because very few studies have attempted to actually link EED to poor wash. This slide shows more or less the summation of these studies, which so far have linked EED to things like mouthing of the soil, to animal exposure, to caregiver hygiene. However, mostly all of these are using small observational studies. Ours, which is also a small observational study, is the first to link EED to poor household water quality. Next slide, please. So we performed a cross-sectional study which was embedded within the context of a larger longitudinal birth cohort study. or the Ugandan birth cohort study, abbreviated UBCS. We assessed EED at the age of 12 to 16 months using the 4 to 5 hour lactulose-mannitol test on a population of 385 infants from southwestern Uganda. Anthropometry and covariate data were abstracted from the birth cohort, in which visits were conducted every 3 months. Water quality in this study was assessed at 6 months using a portable water quality test. In this case, we used the Aquagenics compartment bag test, which detects and quantifies E. coli contamination in 100 milliliter samples of water. Next slide, please. So this table shows the primary water source for each of the households in our sub-study, and they are disaggregated by safe and unsafe, depending on the results of the compartment bag test. Because there is no safe threshold for E. coli contamination in water, the presence of any detectable E. coli in the compartment bag test was considered to be unsafe water. Overall, there was not a statistically significant difference in the presence of E. coli contamination between water sources that are traditionally considered improved and those that are traditionally considered unimproved. With regard to individual water sources, there were some differences in safety. For example, surface water was much more likely to be unsafe, but rainwater, in fact, was much more likely to be safe. Overall, these results make us question how we measure WASH variables in our studies and emphasize the need to actually measure contamination levels rather than using source as a proxy indicator for this. Next slide, please. Furthermore, in this study, we found that children from households with safe water had significantly better lactulose-mannitol ratios and growth outcomes compared to children from households with unsafe water. Also in this study, we retrospectively linked LM ratios measured at 12 to 16 months to poorer growth at 6 and at 9 months. But a limitation of this study was not having prospective growth data from the time the lactulose-mannitol test was performed. The results of this study have been published in the American Journal of Tropical Medicine and Hygiene if you would like more information. Next slide, please. So the next topic is what is the relationship between EED and linear growth in infants and young children in Uganda? So this is the map of the UNICEF's 2019 State of the World's Children Report. Overall, this map shows that stunting remains an enormous public health problem, which most of us know. Globally, about 1 in 5 children under 5 years of age is considered stunted. However, the rate is much higher in South Asia and Sub-Saharan Africa, where NIL mainly works, where it's about 1 in 3 children. It's about 29% in Uganda, where these studies were conducted. While the rate has slowly declined over the last few decades in Sub-Saharan Africa, the absolute number of children who are stunted is actually increasing, mostly due to population growth, although of course now we have to worry about COVID's impact on these rates as well. Next slide, please. So just by way of reminder, this schematic summarizes the 2 main routes by which we believe EED may contribute to malnutrition, specifically poor absorption route and the systemic inflammation route. On the systemic inflammation route, we at Boston Children's Hospital have been focused on microbial translocation markers, specifically anti-flagellin and anti-LPS, IgA and IgG. Next slide, please. So we performed an additional cross-sectional study of 548 6-month-old infants who were enrolled in the same Ugandan birth cohort study. We measured anti-flagellin and anti-lipopolysaccharide at the Gurwitz Laboratory at Georgia State University. Also in this study, we measured systemic inflammation markers, specifically AGP and CRP, as well as markers of iron status, including hemoglobin, soluble transferrin receptor, and ferritin. We assess associations between these markers and infant growth using adjusted linear regression analyses. Next slide, please. So overall, in adjusted models, which included adjustments for systemic inflammation, we found a significant association between higher anti-flagellin and anti-lipopolysaccharide IgGs and reduced growth at 6 months of age, specifically length for age z-scores in our sample of 548 infants. We also found an association between higher systemic inflammation markers, both AGP and CRP, and reduced length for age z-scores. We did not, however, find an association between any of these markers and either weight for age z-score or weight for length z-score at 6 months of age. Next slide, please. We also found a relationship between higher anti-flagellin and anti-lipopolysaccharide markers and reduced iron status, specifically lower hemoglobin levels and also higher STFR. The significant relationship between EED biomarkers and higher ferritin levels was odd, but we believe this may have to do with an incomplete adjustment for inflammation. Next slide, please. And these graphs just show the correlation between the different anti-flagellin and anti-LPS IGs, which are present on the x-axis, and hemoglobin levels, which are present on the y-axis. In all cases, you can see the significant negative association between the two. Next slide, please. So for more information on this study, it can be found in our manuscript, which is published recently in the Journal of Nutrition. Next slide, please. And the last study that I want to mention is one looking at maternal EED and birth outcomes. So the objective of this study was to assess the relationship between maternal EED during pregnancy and adverse birth outcomes, primarily birth length. We performed a prospective cohort study in the town of Mukono, Uganda, located 20 kilometers outside of the capital city, Kampala. And we measured EED in this case using both the urinary LM test as well as serum anti-flagellin and anti-LPS IGs. And we assessed relationships in 258 pregnant women using unadjusted and adjusted linear regression models. So this study consisted of 4 distinct visits. There was first an enrollment visit conducted at Mukono Health Center 4, where a venous blood draw occurred. Samples from this blood draw were sent to the Gurwitz laboratory for analysis for anti-flagellin and anti-LPS. Also at this visit, we conducted an ultrasound to have a more accurate measurement of gestational age. Roughly a week after the enrollment visit, a lactulose-mannitol test was performed on the women. And at delivery, infant anthropometry, including length, weight, and head circumference, was collected within 48 hours of delivery. Next slide, please. So this graph shows the correlation between maternal anti-LPS IgG specifically, which is on the x-axis, and infant gestational age at birth on the y-axis. Again, we considered a big strength of the study to have ultrasound data and therefore more accurate gestational age data. And as you can see, we found a significant negative relationship between the two, suggestive that microbial translocation in particular could be associated with preterm birth. Next slide, please. And finally, in both unadjusted and adjusted analyses, we found that higher concentrations of anti-flagellin IgG and anti-LPS IgG in particular were significantly associated with shorter length of infant gestational age at birth, lower length at birth, and lower LAZ at birth. Next slide, please. And so more information on the study can be found in the manuscript, which is published in the American Journal of Clinical Nutrition. So by way of wrapping up, some conclusions. Within the field of EED, a number of key challenges still persist, including a lack of an agreed upon case definition and diagnostic criteria for EED. Given that EED has the multiple domains that we discussed, potentially a multiplex panel of biomarkers may be a promising path forward to establishing an agreed upon way of assessing EED in the field. The connection between WASH and EED is still hypothesized, but it does have strong biological plausibility. Notably, WASH interventions often do not provide sufficient protection from environmental contamination to prevent or to ameliorate EED and to improve growth outcomes. This was essentially the takeaway messages from the WASH benefits in the SHINE trials. There is, however, mounting evidence that EED impairs linear growth in infants and young children in low and middle income countries. However, more research is needed on the role of EED maternally when it comes to birth outcomes, in infants when it comes to micronutrient deficiencies, and on other forms of undernutrition. So that is a segue into Akriti's presentation, and I will wrap up here. I look forward to your questions, but otherwise we'll turn it over to Akriti. Thank you very much.
Thank you, Jackie. So the title of my presentation is Environmental Enteric Dysfunction During Moderate to Acute Malnutrition in Sierra Leone. Next slide, please. I'd like to take a few minutes to talk about why we studied EED during moderate to acute malnutrition. So globally, it's estimated that there are about 50 million children with acute malnutrition or wasting, and 2/3 of them have moderate to acute malnutrition, which is defined as having a weight for height or length between -2 or -3, or mid-upper arm circumference between 11.5 and 12.5 centimeters. MAM is a public health concern because children with MAM are 3 times more likely to die than children without MAM, and these children are also at risk of progressing to SAM, or severe acute malnutrition, which has a much higher associated risk of mortality. In food insecure contexts, MAM children receive supplemental food as treatment. However, not all children who receive treatment recover, and program success rates have been reported to be between 50% and 100%. A number of risk factors could be alluded to for contributing to these low treatment success rates, one of which could be environmental enteric dysfunction, or EED. And while EED has not been studied among children with MAM, studies by Campbell and Faras have reported that EED is present among children with SAM. And in addition to looking at the role of EED in treatment outcomes for children with MAM, it's also worthwhile to see if there is an association between EED and linear growth among children with MAM, because majority of children with MAM are stunted and they continue to grow poorly even after receiving treatment. Next slide, please. So we heard from Jackie's presentation that there are a number of different biomarkers of EED, such as the LM markers and also the serum markers that she used in her studies. In addition to these, there are also non-invasive fecal markers, and examples are fecal proteins and fecal mRNA transcripts. Common fecal protein markers are alpha-1 antitrypsin, or AAT, which is a marker of permeability, neopterin, NEO, myeloperoxidase, MPO, which are markers of inflammation. And with the mRNA transcripts, we're not just talking about 2 or 3 markers, but we can assess a whole range of markers that can then be used to construct scores. And these can then tell us, most possibly, about a more wide variety of characteristics of EED or domains of EED and what is happening in the small intestine during this condition. So some of the markers that we looked at in Sierra Leone were the gut inflammation score, or GI score, a marker of inflammation, the gut permeability, or gut structure score, that is a marker of permeability, and the gut defense score, which is a marker of antimicrobial gut defense. Next slide, please. So this diagram walks you through the environmental exposures that likely lead to EED, that then likely leads to poor growth. But one area in this pathway that has not been studied as extensively are the changes in the microbiota profile that occur as a result of environmental contaminants. When I talk about the microbiota, I'm talking about microbes that colonize the gut. These could be bacteria, they could be viruses, protozoa, etc. And you may have also heard this term being referred to as the microbiome. Both terms can be used interchangeably. Next slide, please. 2 recent studies have shown that the gut microbiota is altered among children with EED. One study was from Bangladesh, another from Malawi, that then also showed that these alterations are associated with poor linear growth. Among children with moderate acute malnutrition, there are also changes to the gut microbiota. And furthermore, what some studies from Bangladesh have shown is that there are specific foods, if provided to children with MAM, they can lead to growth of more of the beneficial microbiota, so more of the beneficial microbes in the gut microbiota. And while we know that the gut microbiota is altered during EED and during MAM, what remains to be seen is whether these changes are universal across geographic locations. Next slide, please. Therefore, the objectives of the study in Sierra Leone were to examine the associations between different biomarkers of EED and growth during and recovery from MAM. It was also to compare the microbiota profile of children with and without MAM and of MAM children with different levels of EED. And then finally, to examine the relationship among wash conditions, EED, and microbiota profile of children with MAM. Next slide, please. So this study was conducted in collaboration with the Food Aid Quality Review for Foods MAM treatment study conducted in Pujehun district of Sierra Leone. The children were 6 to 59 months of age and were identified as having MAM based on the mid-upper arm circumference tape, and they did not have any complications. They were given 1 of 4 foods as treatment. However, EED was only assessed at enrollment into the program. And we collected urine and fecal samples for this purpose. And fecal samples were also collected from a small number of children without MAM for the microbiota analysis. Next slide, please. So what did we find for the first objective? Next slide, please. Well, first of all, we found a very high burden of EED among children with MAM. This is one of the first studies to report this, using the LM test or the fecal protein markers, for which we have more well-established cutoffs. Next slide, please. And then when we looked at the association between the EED biomarkers and recovery from MAM, which we defined as achieving a mid-upper arm circumference greater than or equal to 12.5 centimeters within 12 weeks of supplementation, what we found was that children with higher GD score, gut defense score, were more likely to recover from MAM, and children with lower intestinal permeability measured using AAT were also more likely to recover from MAM. Next slide, please. Then when we looked at the association between these biomarkers and growth during MAM, what we found was that children with high inflammation measured using the GI score had lower length for age Z-score and lower weight for length Z-score, and that children with high intestinal permeability measured using the GS score also had lower weight for length Z-score. So in this objective, the results show that certain biomarkers of EED are associated with recovery from MAM and growth during MAM. Next slide, please. For the second objective, we focused on the microbiota, and what we found was that, next slide, please, that the gut microbiota of children with MAM, shown in red, was enriched in inflammogenic taxa or inflammation-inducing microbes such as gamma proteobacteria, whereas the microbiota of children without MAM were enriched in the beneficial taxa such as Lactobacillus ruminus. And I talk about taxa because with this analysis, we don't always have information at the species level, so it could be at any of the higher taxonomic levels such as phylum, class, or family. Next slide, please. Then when we looked at the overall microbiota structure of children with EED, measured using the GI score, what we found was that children with lower GI score or less inflammation had more microbial diversity, that was shown in the figure to your left, than children with high GI score. Similarly, the microbial communities between children with low and high GI score was also different, as shown in the figure to your right. And what we also found was that the gut microbiota of children with high GI score were enriched in inflammogenic taxa, such as Streptococcus lutetiae, shown in red, whereas the microbiota of children with low GI score were enriched in the beneficial taxa, such as Roseburia, Blauschia, and Facies. Next slide, please. So basically, with the second objective, what we showed was that the microbiota is altered during MAM and EED. And with the third objective, we were looking at the association between WASH, EED, and microbiota. And what we found was that next slide, please. When we looked at the association between the EED biomarkers and household drinking water source, we first created a binary variable where drinking water source was defined as improved or unimproved based on the UNICEF WHO Joint Monitoring Program definitions. So improved drinking water source would be public tap or tube well borehole. Unimproved would be surface water or unprotected well. And what we found here was that lower intestinal permeability was associated with improved drinking water source of the household using the LM excretion ratio, the AAT marker, as well as the GS score. Next slide, please. And when we looked at the association between the biomarkers and household sanitation facility, this was again defined as improved or unimproved based on the Joint Monitoring Program definition. So improved would be ventilated improved pit latrine or pit latrine with slab, and unimproved would be open defecation or open pit. What we found here was that lower intestinal inflammation measured using neopterin was associated with improved sanitation facility. Next slide, please. And when we looked at wash practices by the household wash conditions, what we found was that more children living in households with unimproved sanitation facility were observed putting soil and animal feces in their mouth. And this was the only practice that was statistically significant. Next slide, please. And in terms of the gut microbiota, what we found was that children living in households with improved sanitation facility were enriched in beneficial taxa such as Roseburia, Collinsella, and a Bifidobacterium species. So to summarize, we saw that certain biomarkers of EED are associated with recovery from MAM as well as growth during MAM, and that the gut microbiota of children with MAM were enriched in inflammogenic taxa, and that children with MAM living in households with improved wash conditions had lower risk of EED and had beneficial microbes. And finally, that the alterations of the gut microbiota are associated with MAM as well as gut inflammation during MAM, which raises the possibility of microbiota-directed or targeting the microbiota with interventions such as nutritional or wash to treat children with MAM and EED. Thank you. And now I would like to hand it over to Shivani for her section of the presentation.
Thank you, Akriti. Good morning, afternoon, evening, everybody. And I'm very privileged to be speaking after Jackie and Akriti, who have done such phenomenal work with their dissertation and postdoc research. And I've been fortunate to be part of Akriti's committee as well as being Jackie's PhD chair. So it's a moment of being very, very proud of both of them. I'm sort of speaking on behalf of another person who she's on the panel, but she isn't able to be online right now, Joanna Andrews Trevino, who has been the third PhD that has been working with Nutrition Innovation Lab on issues linked to our research theme around biological mechanisms. And last week we talked extensively about mycotoxins, particularly aflatoxin, and our work in Nepal around the AFLA cohort study. And so I'm sort of extending, Joanna and I have extended some of the analysis that has been part of the AFLA cohort study to look at EED markers, water hygiene sanitation, but also looking at EED markers and aflatoxin levels in young children. So that's sort of really the focus of this last presentation. Next slide, please. So I don't need to go through the background that Jackie and Akriti have laid out so very nicely. And I realized that I have an error in my first bullet, but I wanted to speak only about Nepal, not Timor-Leste this time. But I think all of you joining from Nepal are aware of the rates of stunting. And that also that the environmental enteric dysfunction, as both Jackie and Jackie particularly laid out, is implicated in the development of the stunting syndrome, if you will. And we've seen that poor wash practices are associated with EED. There's also this postulation that toxins such as aflatoxin may induce EED, and that might be the mechanism by which it increases, it causes poor gut health and possibly poor absorptive capacity in children. Next slide, please. And that's just, I'm sort of using what is Akriti's slide on looking at what is EED, which is a process and a syndrome. And on the top where you see environmental contaminants, I've highlighted the fact that in addition to microbes that are introduced through poor wash practices or through poor wash itself, you have also aflatoxins and other mycotoxins. And of course, this is a postulation, this is a hypothesis, and we're going to try to see what, whether this actually does translate into poor nutritional status. Next slide, please. For those of you who were not on our webinar last week, I do suggest please go and listen to the webinar where we have presented extensively about the AFLA cohort study. But for those of you who weren't there last week, the AFLA cohort study is an observational birth cohort study that was conducted in the Banke district of Nepal. And we followed 1675 mother-infant dyads starting from the second trimester of pregnancy in the woman through almost 24 months age of the child. Next slide, please. So here's just to give you the timeline of the AFLA cohort study, which began in 2015 and finalized its last data collection point in March 2019, which was last year. The launch was, as I said, started with the prenatal in the second trimester of the woman, and then we followed the woman at birth, 3 months of age for the child, 6 months, and so on. Originally, the AFLA cohort study, which you see on the slide as phase 1, was meant to go through the infant turning 12 months of age, and we were fortunate to receive additional funding that would allow us to do follow-ups on the child between 18 months of age and 24 months of age. So what I've highlighted here is the time point that we are able to, we were able to collect additional serum samples to assess mycotoxins beyond aflatoxin, but also we were able to administer the LM test on the infants thanks to the great, thanks to our research manager Ashish Pokhrel, who was extremely valuable in doing this work. And so today what I'm going to be presenting is some of the sort of preliminary analysis that is emerging from that data. Next slide, please. So I'm focusing only on that specific time point, so you have an understanding of the sort of demographics and the descriptives of this population. The child at 18 to 22 months of age in our AFLA cohort study, were about half of them were girls. 20% of them wore low birth weight, and about 7% of them had diarrhea in the past 2 weeks. A lot of them had achieved their minimum dietary diversity, but you can see that stunting, underweight, and wasting prevalences are quite high in the Banke district, as was low head circumference for age C-score. And the last 2 points I wanted to make was the 2 metrics that are often used in programming and in policy around WASH, which is improved water source and improved toilet facility. And what we see is that 96% of the households were reporting an improved water source, and about 64% were reporting an improved toilet facility. Next slide, please. So this is just to give you the descriptives on the EED marker. Now, unlike Jackie and Akriti, we had the opportunity only to collect the urine sample and assess the LM ratio in these kids. We do have serum samples that are that could be analyzed for inflammatory markers as well as for microbial translocation markers, but we are sort of in the COVID lockdown right now with respect to doing any work between the US and Nepal. So what you see here is the LM ratio and the percentage lactulose excreted as well as the percentage mannitol excreted. And you do see that this level of the LM ratio is very similar to what Akriti and Jackie were reporting and is considered on the higher end. Next slide, please. And then this is again, we assess the mycotoxin exposure in these kids, and the first one is aflatoxin, and you do see that it's pretty much the detection rate, and I want to highlight the second column here, which is the most important one for the purpose of this discussion, is that we had about 85% of the kids had detectable levels of aflatoxin. For ochratoxins and fumonisins, there were 100% detection rates, and for DAWN we had 87%. And what you do see across all the aflatoxins that this is they're extremely skewed distributions from very low, a very large number of kids on the lower end of the distribution and a few kids who are on the upper end of the distribution. Next slide, please. So we ran quite a few OLS linear regression models, and what we are presenting here is the relationship of the EED markers. In this case, we're using the lactulose-mannitol excretion ratio, percent lactulose and percent mannitol, and its relationship with wash markers and mycotoxins. In the case of the improved water source, because there was so little variability, there was really nothing we could run in terms of a regression model. In terms of the improved water source, what you see here is that there is a significant relationship between the household reporting and improved water source and the child EED marker LMER ratio. And it's a negative relationship, which indicates that if the child was in a household with an improved toilet source, excuse me, they had lower LMER ratios. And what we did look at was aflatoxins and fumonisins, and we find that there is no correlation between aflatoxins and fumonisins and EED, the EED markers of LMER, percent L and percent M. And all these models have been adjusted for the gender of the child, the number of household members, the mother's education level, and the geographic location, which could be a significant confounder. Next slide, please. So in conclusion, what we do find a significant association of markers of intestinal permeability and absorption and access to an improved toilet facility. And as I mentioned, we don't see it with the improved water source. And we don't, at least with our data, with what we have right now, we don't see any relationship of the LM markers with either aflatoxin or fumonisin. If we are able to assess other inflammation markers as well as microbial translocation markers, I think that would be something we would like to revisit. But at this moment, we are analyzing the associations with growth and development along with EED and aflatoxin markers. Next slide, please. I think, and I just wanted to bring in this particular slide is bring together all the three different presentations that have been done and what this means with respect to research and policy implications. And I think Jackie and Akriti have laid out very nicely what the research implications are. And what we find in the Nutrition Innovation Lab is that our work has allowed us to assess the linkages between WASH, the microbiota, infection health, and nutritional status, and using both stunting and moderate acute malnutrition. What we're hoping is that this sort of advances the thinking in nutrition science and applied nutrition and its intersection with environmental contamination. But what is very, very clear from Jackie's work and Akriti's work is that we need to have longitudinal studies and randomized studies that address the specific issues that have been highlighted. Particularly, and I think there was a little chat on the Q&A about what the intervention was or what interventions are being used for targeting WASH, and that we really need to understand the source, the extent and the intensity of contamination when we are planning responses. The one last thing I would say is that what we have done in these studies has been really focusing on using very innovative biomarkers. And obviously that is something that can be done in the research realm, but in the programming realm, I think the idea would be is that we really need to start thinking about water quality, not just the source of the water, and not just use proxy indicators such as water source and improved facilities, because there could be so many things between that setup of a improved water source and actually the water being consumed that could be the reason why you see such high levels of contamination. So on that note, I'm going to hand it back over to Chris Duggan, who's going to take the next steps in this webinar. Thank you.
Thanks so much, Shibani and Akriti and Jackie for really excellent presentations. And as you can imagine, the chat group has been very active during the presentations. And I think it would be great to kind of address some of the questions that were either left unanswered or can expand on the ones that were in part answered by the panelists and the moderators. So There were several questions that were answered in part, but I guess it would be nice to know if we could review them in more detail. And I'll push the first one to Jackie, who I know you've started to answer this question. A number of our attendees were asking about the role of exclusive breastfeeding in the cohorts that you described. Would you like to comment a little bit on how that was assessed in the field, as well as how it was statistically handled in the analysis?
Yeah, definitely. So because we were looking at infants at 6 months of age for the study linking EED and infant child growth, we didn't have much variability within that variable to kind of be able to control for it. I think about 98% of women reported that they were breastfeeding to some extent at that time. And because it was right at that 6-month mark, it was a time when we weren't expecting some of them to be transitioning away from exclusive breastfeeding. So we weren't going to kind of penalize them for that and make a marker whether or not they were exclusive breastfeeding or not, and the marker whether they were breastfeeding or not did not have much variability. So unfortunately, because we were kind of limited by using data from the birth cohort study for our covariates and not kind of collecting our own more granular data on breastfeeding, we weren't able to do much with that variable. But I appreciate the question. I also am interested in that as a research question as well. I would just think that you would kind of have to work that into the studies that you're designing in order to collect kind of much more specific breastfeeding amounts and levels and et cetera.
Excellent. Thank you, Jackie. Akriti, there were several questions about your presentation as well. Catherine Reeb asked about what are some examples of food that lead to growth of beneficial gut biota.
Sure. So that's a really interesting paper. I hope many of you will read it. So some of the foods were chickpea flour, soy flour, and banana that were able to lead to beneficial microbial, microbes among children with MAM.
That's great, thanks, Kriti. Clemence Mallet had a question about the role of EED in defining or in the definitions that you use for moderate acute malnutrition. Specifically, you used MUAC as the inclusion criteria. If you used weight for height Z-score as the inclusion criteria, would those results have changed much?
We don't know. The program in which this study was embedded used MUAC as their indicator to enroll children in the program, which is why this is what was used in the analysis.
Excellent, thank you. Shibani, one of the questions that was posed was the question about minimal dietary diversity, which was seen in 70% of the cohort of the study that you presented, and that was thought to be quite high. Is that atypical for the population under study, do you think? And could that have affected some of the results?
I just realized I was on mute. So yes, it is something I saw that question as well. It is a little higher than one would see in the Tarai of Nepal, where one of the biggest issues has been that the minimum acceptable diet and the minimum dietary diversity for infants and young children doesn't seem to move very higher than 30, maybe 40%, and I could be wrong. So I think Joanna and I need to really look at why these kids at 8, and it may be that these particular kids were part of the study so that they were getting exposed to messages that led them to have better diet diversity. But the relationship that we are seeing is after we control for minimum dietary diversity. So I think that wouldn't seem to really have an effect. Is that
yeah.
I hope I answered the question.
I think so. To all three panelists, Manola Mazirogas in Guatemala asks about any experience using mycotoxin or inflammation markers in breast milk. Could that provide some clues regarding stunting before 6 months? Any experience or thoughts on that? Very interesting comment.
Chris, I'm sorry, I just kind of missed that. Could you?
Oh, sure. So we've talked extensively today about serological markers and stool markers, but what about breast milk markers for inflammation or pathogens or components of pathogens that might trigger this? Any knowledge there?
Yeah, so I think, Manalo, thank you. I saw your question, and we did, we haven't assessed breast milk for inflammation markers. I think that that's a really important and interesting question. And we have assessed it for aflatoxin M1, which is a metabolite of aflatoxin B1. We found that pretty much most of the breast milk samples did have aflatoxin M1, but it was in very low levels. So it's below what is considered as the EU cutoff for what is acceptable aflatoxin M1 in milk. So yeah, so I think that that's definitely a very good point and something we should pursue if we have banked samples.
Excellent, thank you. Erin Milner is commenting that she says, it's disappointing that most of the studies using WASH indicators are not aligned with post-MDG era and the sustainable development goals. An improved water source is insufficient and not aligned with current evidence. Improved sanitation is also insufficient and doesn't incorporate the negative externalities of poor sanitation from neighbors. Is there a reason that basic water service, basic sanitation coverage, and basic hand washing, are there any plans to reanalyze data using non-dichotomous WASH variables which incorporate these service ladders? Any comments from the panelists on that interesting comment?
I can comment. Yeah, I use the JMP criteria as well, and incorporated the things like distance to water source, and was not kind of seeing an association with what we found in the compartment bag test either. I think that the JMP is noble, and they should be emphasizing a lot more actually testing the water. They do to some extent, but probably don't go far enough, considering I do think that seems to be the actual only way to kind of get at how clean the water source is. I think an interesting finding, we also saw it, that may kind of explain why some things like improved versus unimproved or distance to water source are not doing a great job kind of explaining contamination levels, is that it seems like a lot of the contamination is happening kind of post-collection, perhaps based on kind of the utensils that are being used, whether or not the water is being stored kind of with a cap or not, and things like that, I think are probably kind of underemphasized also in JMP. So it's a good start moving away from just a dichotomous unimproved and improved, but it's still not aligning well kind of what we're seeing when we're measuring kind of actual contamination levels.
Excellent. Thanks, Jackie.
Chris, could I add, or I don't know, Kriti,
Please.
if you had it, you wanted to add something about your WASH work, observational work that you did on WASH, and if you saw anything.
Sure. So we did look in the analysis without using the dichotomous variable. And especially for drinking water, the issue that we ran into was that some of those groups were not balanced. So we had a lot of participants who got their water from tube well or borehole, and very few who got them from an unprotected spring, for example. And it was much more balanced when we looked at improved versus unimproved, but I do recognize the limitations with that analysis. And I think Shabani's question was, did we find any differences with the WASH observation data? So when we looked at household drinking water quality, and using a slightly a proxy measure, because we didn't actually test the drinking water quality, we didn't find differences in how the drinking water was stored among households that source their water from improved or unimproved sources. But when looking at the dichotomous variable, we are finding these differences. I think a next step for this study would be to then go and test the water to see was there contamination in drinking water that was sourced from unimproved and improved sources, and was there a difference? But unfortunately, we did not do that.
Great. Thanks, Akriti. Thanks, Shivani. And thanks, Jackie. Several other interesting questions have come up. Two have the same theme of animal feces. We have a question about, have you addressed ownership and exposure to chicken, and how about exposure to animal excreta and EED? Another question at the same time came through on the same point. So let me ask our panelists about exposure to chicken and other feces.
Is Jackie on? I saw not seeing her.
Yep. Yeah, I'm on. Again, I think we did measure this very carefully, actually, in my first study that I presented on water quality. We did actually collect good data on exposure to animals, exactly kind of which animals in particular, as well as how far they were from the household and things like that. Unfortunately, those familiar with the Southwest and Uganda, again, I don't think there was enough variability. It seemed like children were kind of saturated by animal feces and being around animals. Most households kept animals in their homes. So while we didn't see much, that might just simply be because it wasn't a variable with a lot of variability, unlike the unsafe and safe water, which was split very evenly among households very nicely. And so we got that variability. While we couldn't exactly quantify how much children were exposed to bacteria from feces, it was clear that all children were hanging around animals almost all of the time. So that could be why. But of course, the study that I presented on the slide with showing the studies that have been done linking poor sanitation to EED, there is a study there that I would refer you to linking exposure to animals and EED. So there is some evidence for it. We just did not see it in this particular case.
Excellent. Thank you, Jackie. Shivani?
Sure. Can I add one point?
Please.
I think one of the issues that one faces when you are in an environment, and I think this might also sort of address what Erin sort of pointed out too, that you're in an environment where there is contamination across the board, and that the sort of these across household contamination that happens. And when you're doing a study, even if you have the most rigorous methods of data collection, if you don't see a variation, then you're going to not be able to, if you run that regression, you're going to be like, well, there's no relationship. But in fact, what it is there's very little variation in that population for you to see any difference. And I think that that's what we often run into, which is what Jackie has been indicating. And someone asked that question about improved water source for Nepal, where 96% reported improved water source. And I bet you it's the same issue, that everybody has some form of a water source that they perceive as improved. And we had the list of what is improved and what is unimproved. But I think it's not an individual, it's an environmental and a community level issue that doesn't allow us to differentiate between households in this kind of analysis. So Chris, that was just.
No, excellent. That's great. I have a couple more methodologic questions that I'll bring to the panelists, but I do want to make sure we have time to devote to think about the policy implications of our work, because these are all fascinating analytic approaches. And but let's ask one more kind of global question, which is one of the attendees asked about, did any of the studies attempt to assess levels and types of mother-child interactions? Did improved maternal stimulation make a difference in helping children with moderate acute malnutrition improve neurodevelopment as well as growth? So Akriti, I'll point it to you since it's a question about MAM.
Sure. In my study, we did not, but in FAQR, there is another study on neurocognition, and they didn't look at whether stimulation led to improvements in neurocognition, but what that study did find was that after supplementation, there were improvements in neurocognition.
Great. And similarly, we've done some work in Tanzania that's linked to biomarkers of EED with different aspects of the Bailey scales of infant development, but we haven't formally measured maternal stimulation. So that's a great question. Okay, so why don't we open the door a little bit up to kind of given the science, given the state of the science, what are the policy implications? We're obviously doing this not just to learn about science, but to have a positive impact and develop more broadly based policy for improving growth in young children. Let me push that up to the panelists and say, knowing what we know now, knowing what we've reviewed so well over the past few minutes, what are the next steps in terms of advising people who make health policy in low and middle income countries? Are the data strong enough to say, hey, EED is a major problem in the countries we reviewed that we need, or do we need more data to define it better, correlate it with important outcomes, and perhaps do more intervention studies that would suggest not only do we know how to measure it, but we know how to treat it? It's a tall order. I'll start with Shivani, since I know she's thought about this.
I was hoping Jackie turned off her mic. She was like, off.
I could go. I could also go.
Why don't you lead and start off, Jackie?
Why don't you do it, Jackie? Come on.
All right, I'll start off. Just a few points to make about that. You know, I think we're all kind of coming off in this community, like having done a thorough kind of post-mortem on WASH benefits and on SHINE. And, you know, WASH isn't the most kind of popular thing to think about or to fund right now. But, you know, I would say there is a lot of good evidence from observational studies that point to a strong link between WASH and EED, including kind of what Akriti and I have been seeing. So to not give up on WASH, but to not think about WASH in such a rudimentary terms, that we need to be thinking about WASH, both like what Shivani said at more of a community level and also comprehensively, kind of researchers, we should kind of expect, you know, more from ourselves and better from kind of the communities that we're working with in terms of what they deserve in terms of WASH, and simply having an improved pit latrine does not mean that they have sanitation, and simply having, you know, access to a borehole does not mean they're drinking clean water. So to keep that in mind and to kind of keep WASH, I think, at the forefront would be good. And then in terms of EED, certainly as researchers, we need to kind of settle on our case definition and settle on kind of how we are thinking about measuring this. And there is a lot of work going on, though admittedly every kind of research group has their own biomarkers that they're touting, which is not doing much to kind of advance the field. But I think, as Chris mentioned, it's also probably time to start rolling out better randomized controlled intervention studies. Chris and I did one looking at zinc and found that zinc supplementation, and found that it did not have an effect on improving markers of EED, again, the antiflagellins and the anti-LPS that we had been looking at. So that was, while a disappointing result, it kind of awakened us to the idea while we were working on that manuscript that there just was not a lot of good rigorous intervention studies for EED, and we should probably start advancing kind of the bar on that as well.
Yeah.
Thanks, Jackie.
Thank you, Jack. Sorry, yeah, I was looking at Akriti, but I think I can just jump in and sort of, I think on the research end, we still have a lot to unpackage, if you will, around EED. And I think that's what, this is just a starting point. But what we are seeing is that there is EEDs out there, it's prevalent. It could be correlated in some cases to certain health outcomes, but maybe not others. That does not take away the fact that it is a condition that is pretty debilitating when you're talking about gut health and particularly increasing risk of inflammation. And we can talk about whether that causes poor absorption of nutrients or not, and to what extent it causes, but the fact of the matter is it is increasing the risk of poor health in young children and in mothers who are adolescent women who are going to have going to be the new mothers. So I think that that's one point, and I think that Jackie has sort of really nicely articulated the research needs here. On the policy end, I would say that it's very hard when you are a policymaker or a programmer to think about how to bring these very biological elements of our research into policy and programming. And I think that the first place I would think about starting is to sort of say, and I know that a lot of people have already worked on thinking about the wash indicators, and that just having improved versus unimproved sources, which is something that is highlighted even in our presentations where we are looking at it dichotomously, isn't sufficient to indicate that that community actually is free of pathogens and is free of contamination. On the food end, we are really talking about the food systems approach, and that does bring in food safety. And I feel like we need to start talking more about environmental safety as well. And no matter how many research studies we do, we can go back and forth about this. If there's no variation in a population, we won't see the relationship, but that doesn't mean that those issues are not important. So I would say that thinking about taking food systems and food safety to another level of bringing in environmental contamination and environmental safety around wash. The other thing that I want to point out, that I think that one of the questions on neurocognition and maternal stimulation. So a lot of the work that we've been doing is, we have very focused research questions that need to be answered, but I'm hoping as we're doing these webinars that we're putting the puzzle together with these different pieces. So we have research that has looked at the role of animal source foods, that has looked at head circumference for age z-scores, which could be possibly a proxy of brain development, particularly in young pediatric populations. So I think that there's a lot out there that needs to be brought together. And I'm hoping as we go towards the conclusion of these webinar series that our charge as a Nutrition Innovation Lab will be to bring all these pieces together for folks out there. And I really appreciate all the questions, because that's making us think a little bit more deeply on what we want to bring together as, sort of here's what the output of the Nutrition Innovation Lab is. So that's, on that note, I'm going to pass it back to you, Chris.
Excellent, thanks. Thanks, Shivani and Jackie. And Kriti, I see your hand up, so please.
Thank you, Chris. I do have a comment. I think what my work in Sierra Leone has shown is that we've really added to the evidence base, especially around thinking about not just, what impact do these interventions, nutritional wash, have on anthropometric outcomes, but also pushing us to think more about the gut microbiota, because perhaps this is where we need to be focusing on and using the gut microbiota and different taxa as targets for these interventions. And, for program implementers, the science isn't there yet where we can say you do need to promote consumption of banana or soy flour that one study showed. But this is where the science is heading. And I hope that in the near future, we will be able to make those recommendations.
Excellent, thank you. I'm reminded, of course, in terms of, I hope everyone, and I'll add it to the chat group, was able to view last month's really interesting paper in the New England Journal, where Jeff Gordon, Tamid Ahmed, and colleagues did some fantastic work about how the gut microbiota was influential on growth of young children. And I do know that the Gates Foundation has taken up with moderate enthusiasm this idea of microbiome-directed complementary feeding. So I believe that'll be very high on the research agenda over the next months to years. Patrick Webb, you've been awful quiet. I wonder if you had any comments on the policy implications of the work we've heard about today. I know you're handicapped a little bit by IT support today.
Definitely handicapped. All right, well, thanks, Chris. I think it's important that what Akriti just said, these studies have been focused on pushing forward our understanding through the science of links, let's say how various how environmental enteric dysfunction can be measured to begin with, how that links to diet and water quality, and how each of those link to various kinds of outcomes relating to nutritional, not just wasting, but linear growth and micronutrient status. Obviously, what we've presented is just the tip of the iceberg. What we were not doing was evaluating WASH interventions. I think that needs to be spelled out. And I think we need to do a lot more of both. We need to continue to push the frontiers of our understanding of what EED means for these children physiologically and in terms of growth and nutrition and future health. And where are the entry points for intervention that can make a difference? And if, in fact, and I saw Felicia Wu was asking in one of the questions, if appropriate sanitation and clean water at water sources are not sufficient, then what are going to be the additional activities that governments should be promoting and development partners should be focused on? Communications, obviously better practices, better understanding of what exclusive breastfeeding means and not feeding contaminated water to infants. You know, there are lots of other dimensions that come under the rubric of policy. But we need to speak clearly to the policy makers, the governments in these countries, to communicate where our understanding is going and what kinds of actions are feasible, and then test them in the field. I think all of those things need to be done. That's where we're heading. And I think that's what these kinds of studies contribute to.
Excellent, thank you. Thank you, Patrick. Scrolling through the questions, I see many have been answered not just by the panelists, but by participants, which is terrific. And a question has been raised about the, and Akriti, I'll ask you to comment perhaps on the WASH interventions, because one of the attendees was wondering whether the WASH interventions didn't include chlorine, and how could that affect perhaps the absence of effect on cryptosporidium. And I'm sorry, maybe that was Jackie that reviewed some of the lack of efficacy of the WASH intervention. So either Jackie or Kriti, any thoughts on that?
My study did not was not focusing on the WASH interventions.
Right. And Jackie, any thoughts on your review of the literature for WASH and whether cryptosporidium is an important factor in EED? And while you're thinking about that, I guess I will address another comment that came up in the chat, which is this idea that, of course, the antibodies that your work has often used are indeed antibodies to gram-negative cell walls and components, and therefore leave open the possibility that they don't really explain any of the exposure to other organisms, including viral organisms, including gram-positive bacterial organisms. And furthermore, you could argue that the presence of these gram-negative antibodies to gram-negative components that are found in the bloodstream, we assume that they're related to gastrointestinal exposure of gram-negative bacteria, but there are other mucous membranes that indeed these products could be absorbed from, as well as the skin. So I do think that a note of caution is warranted for when we think about these as truly EED biomarkers. So yeah, Jackie, any thoughts on cryptosporidium as a possible cause of EED?
Yeah, I think this was more measured, I think, in kind of WASH benefits. So I would probably direct you there. What we did was, because E. coli is probably like considered more most common water contaminants that you see. We kind of used it as like a proxy marker for perhaps how contaminated the water is with both bacteria and parasites, which unfortunately was just because we did not get kind of the water tested for that. But yeah, I think going forward, maybe I would heed my own advice a little bit and not just look at E. coli and actually kind of exactly what the water is contaminated with, including Cryptosporidium.
Excellent, thank you.
Mm-hmm.
One question was posed about the role of probiotics. We think of EED as an entity where bacterial enteropathogens are present in the gut and cause mucosal damage. But what about the role of, quote, healthy bacteria? What are the panelists' thoughts on that?
Sure, I can talk about the one study that looked at whether giving Lactobacillus GG to children 3 to 5 years in Malawi had an impact on EED, and they found that it did not. My response to this would be that perhaps the age group was slightly older and we need to look at a younger age group because we know that EED peaks around 12 months. And perhaps we also need to be looking at other probiotics, so in addition to Lactobacillus GG. And so this is where focusing on what are the beneficial bacteria that are present in children without EED might be helpful.
Excellent, thanks, Akriti. Jackie?
Yeah, I mean, just to build off that, yeah, the probiotics study was, a negative no result, as was our kind of zinc, RCT, the antibiotics ones have been as well. So there have been EED therapeutic trials, mostly kind of small, but I don't think there have been any ones that have shown kind of any positive impacts on EED, which is kind of why I tend to go back to kind of the WASH question. Not only is it a prevention, but I think in terms of the therapeutics that we have tried and have not shown any positive results, we perhaps need to kind of go back and start thinking about prevention and WASH while keeping open, further therapeutic trials, but just kind of to point out that we have not really seen much success in the ones that have been tried thus far.
Excellent. So I'm going to keeping my eye on the clock, I'm going to ask our 3 panelists for a little bit of a wrap-up comments on both their presentations and the discussion to date. And maybe we'll go in the same order if that's okay. So Jackie, any wrap-up thoughts?
I've loved the discussion. Always great to kind of talk about what we're measuring, how we're measuring, if we're actually measuring what we're talking about. I think kind of very important things to keep in mind. Chris and I have like a lot of in the pipeline when it comes to EED. We're very excited to work with PATH kind of to develop maybe a multiplex assay that's looking at various domains of EED. And I know Kriti has exciting work on metabolomics and proteomics, which is also, I think, kind of a future direction this field is headed. So there's tons of exciting work within EED if you're thinking about kind of getting involved in it. It's a super interesting field that still needs a lot of people kind of tackling it from all different angles. Looking forward to any future collaborations with anybody out there who may be interested.
Excellent. Akriti, any comments?
Sure. We've studied EED and we've used a number of different biomarkers, and we've found that a lot of studies say that we've tested interventions, WASH, nutritional, and we didn't find that it improved EED. And my response to that would be, let's think about how we are studying EED, which biomarkers we are using. I think the mRNA transcripts might be, a comprehensive way to study this condition. And in addition to using the biomarkers, let's think about the age group of the kids. Some studies have looked at older, some have looked at younger, but I think when we measure EED is going to be important. How we measure it, the study design. So I hope studies that continue to work on EED and looking at the association with growth and looking at which interventions can improve EED, think about how EED is measured and the age of the study participants and the design of the study.
Excellent. Shibani?
Thank you. And I think what I'm very excited about is from this webinar and the previous ones is that we've generated a lot of discussion and we've got such fabulous attendees here who should all actually, some of them I wish we could have them speak to us and with such great ideas and sort of thoughts moving forward. In terms of the from my perspective as an applied researcher, I think what I'm really excited about is that we're able to take nutrition science down out to applied nutrition, but we're also moving it towards folks who are working in programming and policy. So that I think has been a very interesting journey for me within the context of this work that has been conducted around EED and the biomarkers of EED and WASH and nutritional status. I do think that we are moving forward in different studies to look at interventions which are more sort of programmatic interventions that could then be measured using innovative markers to see what within the WASH realm, what within the realm of improving practices in the household, animal-livestock interactions could maybe shift the marker for EED and for nutritional status of those populations. So there's a lot more going into more of the interventions realm as we move forward. And so this is, for me, this is a starting point, but it's been an exciting journey and I'd like to continue with all of you on this. Thanks so much.
Thank you, Shibani, Akriti, and Jackie for really excellent presentations. Thank you for the Nutrition Innovation Lab for supporting this seminar, as well as our colleagues at USAID. I've learned a lot. We need more tools to measure EED and impact, as Shibani noted, for therapy. So let's keep the conversation going. Please visit our website. Keep us up on your progress, and we'll do the same. So thanks very much to all who participated today, and see you soon.